And let me introduce. So where is it? Oh, give me a second. Sorry. Okay. Okay. So hello everyone. Welcome to the first talk of our fall colloquium series. First speaker is Professor Lei Dai. Dr. Dai is currently a principal investigator at Shenzhen Institute of Advanced Technology and also the director of the Center for Synthetic Microbiome. His academic journey began with his BS in physics from the University of Science Technology of China, followed by a PhD in physics from the MIT. After completing his doctoral study, Dr. Dai became a Jane Coffin Child Postdoctoral Fellow at UCLA. His groundbreaking research focusing on ecology of complex microbial community, utilizing experimental and computational approach to improve host health through precision microbiome engineering. Dr. Dai has been recognized with several prestigious awards, including the American Physical Society's Outstanding Thesis Research Award and an MIT Technological Review's Innovative Under 35 in China. We are fortunate to have him share his insight today on the cutting-edge field of microbiome engineering, a field poised to revolutionize health, agriculture, and environmental sustainability. Please join me in welcoming Dr. Lei Dai.
Okay. Thank you, Professor Kim, for the very kind introduction and invitation. It's a great honor to speak at IBS Biomedical Mathematics Colloquium. And today I would like to share with you some of our recent works on quantitative ecology of host-associated microbiomes. So what my lab is interested is in using mathematical modeling and our quantitative tools to understand the ecology of the complex microbial communities, and specifically those communities that reside on animal and plant hosts. A lot of you probably have heard about host-associated microbiomes, but I would like to briefly introduce the concept because I know a lot of you are from the mathematics and physics background. So the microbiome actually is a emerging new field that I think probably like around 20 years ago with the emergence of cheap next generation sequencing that people started to go around to different places on the earth and extract the DNA and sequence what's in there, right? And what you can see here is I think a milestone paper of the Earth Microbiome Project, where people sequenced hundreds of different sites on Earth and reviewed the great diversity of the great microbial diversity. So what people found was that there are hundreds of coexisting microbial taxa in the animal gut. And also in the plant rhizosphere. So, for example, on the left is called host associated, and left is free living, such as soil and water. And the y-axis is actually, you can just treat it as the number of species or taxa. And so the highest is the plant rhizosphere, which is the area close to plant roots, and also the animal distal gut. So it's there's a huge diversity. And also these on this principal coordinate analysis, you see that for different niches, there's different set of assembled microbial communities, and apparently they adapt to that type of ecological niche. So it's within the past decade, there's a lot of breakthroughs, and people start to realize that these microbial communities have a lot of relevance for host health. So for example, for human, the gut microbiome, and as well as other microbes on other sites like oral and vagina, they have relevance for a lot of important diseases, and also for other animals. And with worms to rumen animals. A lot of animals, they have their commensal bacteria, which provide important functions such as breaking down polysaccharides and defend the host from the infection of pathogens and detoxification, just to name a few. And also for plants and the microbes residing on the leaves and also close to the roots, they provide important functions. They help the plants to deal with biotic and abiotic stresses. So it's really, I think, an interdisciplinary area where I think people coming from, for example, the biomedical fields, from the agriculture field, start to realize that microbes also play an important role for the eukaryotic host. So like I mentioned in the beginning, I'm interested in microbial ecology. So just like in physics, where you first ask what makes what are the compositions of matter, right? So you look at the atoms and what are they made of. And for microbiomes, what are they made of? They're made of different microbial species. And basically microbiologists classify them into different taxa. So at the level of kingdom, like we have bacteria, archaea, fungi, and also many viruses, including eukaryotic viruses and bacteriophages. But what's more is that we need to understand the interactions among these microbes with each other and with the environment, right? So there are different types of interactions, and maybe it could be neutral, but it also could be mutualistic, where A helps B and B helps A. But a lot of times you also find a lot of competitions. A inhibits B and B inhibits A. So these different types of interactions really shape the ecology of the microbial communities. And a lot of times I've also been asked like why microbial ecology matters, right? So it's not just for curiosity. Actually, for host microbiome, they play important roles. For example, this is a slide I took from the United States Center of Disease Control, where it's clearly stated that the microbial ecology is a field where we help us understand how germs influence health, keep balanced microbiomes, and protect people from infection. So it's important to have a balanced, healthy, commensal microbiome to reduce the probability of being infected by pathogens. And so this is one very clear point. And another point is that when we were born as babies, and we actually started from a germ-free state, and then we got exposed to microbes from our parents, from the environment, and there's a community assembly process, very much like a rainforest, where if you started with a fire, there's nothing, and then you have some small plants, and then the big plants, and then there's an ecological succession process. So in our gut, there's actually a succession process where some microbes got dominant and then being replaced by others, and it's a very interesting community assembly process that over the first 3 to 5 years, we started to have a mature gut microbiome. So what really matters is that, so we need to understand microbiomes from a system biology perspective. So it's not just a set of independent living microbial species, but they interact with each other. So last year in Cell System, there was a special issue focused on microbial communities, and I took this cartoon from Professor Yang Liu's perspective, where he showed that using a network, you just treat each bacterial species as a node, and you illustrate the interactions, facilitation or inhibition using these edges, and there's a directed graph, and then you can try to control the nodes to, for example, increase the growth rate or inhibit the growth rate of certain species. And the goal sometimes, for example, in this case, he put the C. Clostridium difficile, a well-known pathogen, in the middle, where the goal might be how can I reduce the bacterial load of C. difficile, right? Well, we've given this network. So it's a system biology perspective where similar to, I think, the system biology at the molecular level, where people start realize that, okay, we need to control the understand and control the protein-protein interaction. So in this case, the microbial interspecies interaction really matters if we want to understand and control the function of the microbiomes. So in this talk, I want to illustrate some of our efforts on quantitative microbiology. So the first two stories are focused on ecological dynamics, and then I will show you how we develop some quantitative novel methods to understand the strain composition as well as the spatial structure of the microbiomes. So okay, here we go. So the first story published in the ISME journal, where we used lots of mathematical models to infer key ecological drivers and interactions in the microbiome. And I just show you some of the results. So as I said, ecology plays an important role in community assembly. So around the time that we published our paper, there was a Nature paper, also a very cool one, about the multi-kingdom ecological drivers on microbiota assembly in preterm infants, where you can see that this color from white to black illustrate the trajectory of the maturation of microbiome within the first year of the infant. And these colors, yellow, red, green, blue, illustrate the dominant genus at different time points. In this paper, the authors try to use the Lotka-Volterra model to infer the interactions, inhibition, and facilitation to understand why certain species are dominant at certain time points. So this is a very interesting ecological observation and try to the understanding. And we actually started this project also around 2019, and we were actually looking at a different phenomenon. So in adults, when we know that the microbiota is quite stable, unlike infants, but when we have dietary interventions, for example, if we intake some dietary fibers like inulin, resistant starch, that basically they are oligosaccharides that cannot be utilized by humans, but can be utilized by the gut microbes. So and then it's known that when you intake these fibers, the gut microbiota will change, got perturbed. But what we don't really know very well is that so for different individuals, they might have different responses. Okay, so some individuals have strong response and some not. And we also had another independent study where we have a small cohort, around 30 people that consumed inulin and observed this individualized phenomenon. So what we're trying to do is to use ecological network to understand what's going on. And so I just show you this set of data in the murine gut microbiome. So we have a control group where we fed the mice with cellulose, and for the experimental group, we fed them with inulin. Okay, and then we collected fecal samples at different time points. So it's a time series, and what you can see that the microbial diversity. So here we use Shannon index. So basically, you can think about it as the more diverse means more number of species and more balanced microbiome. So within the first few days, there is a rapid decrease actually in the alpha diversity of the microbiome of the fiber. So what happens is that some species got really dominant. Okay, they respond to inulin and they grow very well. So the diversity goes down. But then in the time frame of like 4 weeks, they gradually reached a new steady state. Okay, and you can also look at the how the microbiota, the composition shifts over time, like a trajectory. So that's actually our observational data, right? So what we're trying to understand what's going on. So we used a model, a well-known ecology model.
Late in your previous figure, why do you compare the different city? Because it's interesting, the Beijing, Hunan, so Guangdong and Shanghai.
Yeah. So different panels are actually mice. So with different geographical like baselines, and so Beijing, Guangdong, Hunan, Shanghai, they are actually mice from different vendors, and these mice, when they were born, they had different setup. baseline microbial communities. So because we want to study how different baselines might affect their response. Actually, you see where their response is quite distinct. Yeah, so different mice and yeah.
I see. Interesting. Okay.
Right. Just to have more baselines, then you will see
Mm.
different types of response, right? Yeah.
Mhm.
So that mimics what happens in humans. Yeah. So if we apply the time series, you have a time series, then you can fit the time series using you can use the time series data to fit the generalized log of a Terra model. So in this model, it's very simple. So originally, ecologists, mathematical biologists use that to understand why there's oscillation, the predator-prey dynamics. And so in this case, we have a basal growth term and also have an interaction term. And we also introduced a term that we call a fiber response. So we assume that when you introduce fiber, it stimulates the growth of certain bacteria, taxa. Okay, and so applying that to our data, time series data, what we inferred was that some species, some taxa, for example, Bacteroides cystophagens, Muribacteriaceae, just name a few. These are the taxa that have a positive epsilon, a positive fiber response. And we also infer the beta IJ, so the interactions among these bacterial taxa. So you can illustrate that using a network. Okay, so not just we know which species respond to inulin, but also how they interact. Okay, so then we can try to understand what's going on there. So for example, what we see is that these purple purple lines are the primary degraders, so the species that their growth rate is stimulated by the inulin, so they have a very strong response. But also there's the other species, for example, I can mention Mesonephila, which is like they respond a little bit later. And but what happens is that, for example, for different baseline from different regions, so the Hunan, the baseline is actually because they have a higher baseline abundance of this taxa, so they respond, but others don't really respond. Yeah, so it's kind of like the baseline abundance kind of determines their response. And it's more evident, for example, also you see that for different baselines, because there's competition between these degraders. For example, in the a lot of times what you see is that the Bacteroides cystophagens grows up and got outcompeted by the Muribacteriaceae. So because this red taxa is actually a very strong matters. And okay, I forgot to mention, for example, in Shanghai group here, because they lack the responders, their absolute abundance of microbes is actually compared to other baselines is quite low. Okay, so I think these illustrate that these different baselines, because of their difference in the microbial composition, they have that influence their response to inulin. But really, I think we want to validate some of the inferred interactions and key fibers. So we perform some extra experiments, and I just show you some interesting results. So the one is that we apply this model to a synthetic gut microbial community. So in a previous published paper, they introduced a 15-strain consortia to germ-free mice and also 14-strain consortia to germ-free mice. And using that data set, we inferred that there are strong competition between Bacteroides vagatus and Bacteroides feingoldii. So our prediction is that so there's B. vagatus inhibits B. feingoldii. Actually, in the time series, what you see that in the absence of B. vagatus, B. feingoldii can grow up very well. So we actually isolated these two bacterial species from the healthy human gut, and we did in vitro co-culture. What we see is that in monoculture, this strain grew very well, but in co-culture, it was completely inhibited by the other strain. Okay. So that's kind of a key validation, right? And we also, so this is a simplified, like 15-strain consortia, we applied this to a natural human gut microbiome. So there's a cohort about 10 time points and with baseline phase as well as the post-intervention phase. And we inferred that, for example, first thing is Bifidobacterium adolescentis is a responder. And this is well known actually. In your yogurt, for example, you will find this probiotic B adolescentis together with inulin because the prebiotic will stimulate the growth of the probiotic strain. What people didn't know before is the ecology, right? So in this case, we inferred that this responder is a strong inhibitor of the Bacteroides decoris, and we also validated that in co-culture, pairwise co-culture, and we also introduced these probiotics into the feces of three different persons, and we indeed find that 2 out of the 3 times there was a strong inhibition of the Bacteroides. Okay, so this is just to show that now we can understand which species respond to the prebiotics, and also we know what's going to happen for the network, right? So for the other species, and we can try to use the model to forecast for different individualized baselines what will happen, right? So if you intake inulin or other prebiotics, how the gut microbial ecology will shift over time. Okay. So this is the first story where we use the generalized Rotaro model to infer the key interactions among the microbial species. So the second story published earlier this year is that we look at a different perspective where we use data-driven models like machine learning and deep learning to understand colonization resistance of the gut microbiome. Colonizing resistance is another, I think, very interesting and important function of the microbiome. So this is a definition taken from ChatGPT. So it says colonization resistance refers to the ability of the indigenous microorganisms within a host to prevent the establishment and growth of potentially harmful pathogenic microorganisms. So, for example, if you look at the illustration on the right, we already know that the commensal bacteria protects us from the infection of pathogens. For example, for C. difficile, there's commensal bacteria called Clostridium sindens that produce secondary biosalt to inhibit this pathogen, and there are other mechanisms as well. So it's very important to have, like I mentioned, to have a healthy and balanced commensal bacteria. Then they have different molecular mechanisms to inhibit the pathogens. And to understand the microbial colonization resistance actually has a lot of significance. For example, we can utilize this phenomenon to decolonize pathogens. And on the other hand, we could also in some way to help us to improve the colonization of some beneficial microbes. For example, in agriculture where you have fertilizers, but a lot of times these microbial fertilizers may not colonize very well in the soil, right? So there are 2 sides of the stories. So for human microbiome, there's very interesting phenomena that just like the per individualized response to prebiotics. So there's individualized response to the infection of pathogens. And so there's a Cell paper that find that there's interpersonal variations among the susceptibility to cholera infection. So we know that Vibrio cholera is a microbial pathogen for cholera. And among different individuals, so from left to right, they're different persons. Their infection of cholera have a huge variation in the bacterial load of this pathogen, so over 1 to 2 orders of magnitude. And on the other side, like I said, for probiotics, there's a similar phenomenon. So for the question?
Oh, no, it's fun. Yeah.
Oh, okay.
Yeah.
Probiotics. So I continue.
Yeah.
Yeah. So probiotics, then, so they're healthy volunteers that they took probiotics, and so different participants, and these colors correspond to the load of the probiotics. Actually, at different sections of their gut. So in this Cell paper, they also find that there's a huge variations among different participants, right? So this also, I think, for some of you, you might have taken probiotics and you may have like a good feeling or bad feeling or no feeling, but so it's individualized, right? So because for some people, the probiotics colonize and then they can express some beneficial function, but others, they just don't live, right? So they're dead. So the challenge, I think, is to, based on the huge variations among the gut microbiome, can we predict and manipulate colonization resistance? So the traditional microbiology approach, I think, is becomes infeasible for complex communities like the gut microbiome, around 100 species. So like I said, microbiologists usually start with understanding the molecular mechanism. So there definitely a lot of interesting mechanism like type 6 secretion system, also the like short-chain fatty acids. There's just so many of them, but we have very limited knowledge of diverse process governing the microbial interactions. So if you have 100 species, then you have like around 5,000 pairwise interactions. So it's just impossible to figure out all of them. Can we actually make system level prediction without understanding the mechanistic details? So we started with this question, and then we found a solution. So it's not, I would say, a completely new solution, but I think well, I want to draw an analogy to the protein field where there's because I was trained as a physicist, I think we all know that the protein folding is a very important biophysics problem. And for 50 years, physicists wanted to solve this problem, but with some success, but not much. But the AlphaFold AlphaFold 2 specifically really had a huge breakthrough in predicting the structure of the proteins from the zero sequence, right? So it's basically because we accumulated enough protein structures in the PDB database, then we can train this deep learning model to predict the structure. So what we wanted to do is we actually use data-driven approach to study microbiology. And if you think about the mathematically, there's analogy, right? So what we try to do is we try to figure out, okay, if you give me a community composition, it's the basic high dimensional vector, say 100 to a few hundred different species, right? So I know their composition, and there's some network, but I don't really know the parameters. But what I'm trying to figure out is like what's their function, right? So if I have enough data, maybe I don't need to know like the mechanistic details. So in collaboration with Professor Liu.
Lei, in your case, is how the usually typical data looks like from the one person? It's a matrix or vector. So I guess different type of the microbiome and then their level, or what kind of data do you get typically?
Oh, so we get actually just with in a real number. Like for example, I have a vector, and eventually what I'm trying to predict is a real number. Let's say what is the number of colon CFU of the pathogen, or what is their production of butyrate or specific metabolites, for example. So basically a mapping from a vector to a number. Yeah, but could also be a vector if you say I try to predict multiple metabolites at a time, right? So it's not a matrix, I say. Yeah.
I see. So anyway, raw data is a vector. So vector is composition of the various microbiome of each human.
Right, and sometimes we talk about absolute abundance, so it's not just let's say a compositional vector, but also could be the absolute load of each taxa. Yeah, so right. So it's a vector, but it does not has to add up to one. Yeah. Okay. Yeah. So it's a vector. Each person's gut microbiome is represented by a vector, and then you try to predict how good is that vector, right? Is it a protective microbiome? Is it a microbiome that can produce the enough butyrate, for example? Yeah, so something like that. Yeah.
Yes. Okay. Thank you.
Yeah, okay. So yeah, so this paper, I think this is kind of like a very short abstract of this paper. So we can treat all the gut microbes we have like as a meta communities, right? And then each person is a local community. So if you say all the people attending this colloquium in total have 500 bacterial species in their gut. And each one of us is the sampling this meta community that we have, let's say I might have 120 and you have another 100 species. So, but they have some overlap, but not but they were different, right? So the each is a local communities, and because of this local community has a distinct set of species, then when we got invaded by an exogenous species, it could be a pathogen, it could be a probiotic, then there are different outcomes. So sometimes the exogenous species got colonized, so they invade successfully, and we call this outcome permissive. And sometimes it invade but quickly disappeared, went extinction, so that's resistant. So there, qualitatively speaking, there are two different outcomes. But for the ones that are permissive, so detectable, either by qPCR, by sequencing, okay, so at a new steady state, it's existent, then also they have different levels of relative abundance, right? So some permissive communities after colonization, there's high abundance of the rat species, some of the low abundance, right? So what we try to do is that give me the community structure, the vector. So I would train the model based on the observed colonization outcomes. And then using my test data, I will try to predict, so qualitatively and also quantitatively. Okay, and then I can also try to figure out what species out there actually contribute to the colonization resistance, right? So that's kind of like the overall story. So before I show you the experimental data, I just want to just very briefly show you how we got inspired that we think the data-driven approach is feasible. So if you believe that our community follows the generalized Log-Terra model, which is not really the case, but it's a good model to start with, then you can do some analytical derivations to figure out what is the pre-invasion abundance as well as the post-invasion abundance of the different species. So what we found was that for the exogenous species, so that we call S+1, this new species, when they come into this a resident community, we can, in Log-Terra model, you can actually derive the post-invasion abundance, and it depends on these different parameters. So one is the abundance of the resident species, and also the interactions of the exogenous species onto the resident species, as well as the interaction of the resident species onto the exogenous species. So these are vectors, and A is a matrix, that's the interaction among the resident species. So basically, this is a very elegant derivation that just tells you if you have this pre-invasion abundance, right, then that's the vector I was talking about. And this is the real number I'm trying to predict. Right, so analytically, if you believe the world follows the Log-Terra model, then this is the relationship. And what it says is that actually, so without knowing the parameters, you could do a linear regression, and then you can have some reasonable prediction, right? So you don't need that much of data. And this is just to show you that the analytical prediction is consistent with simulations of the Log-Terra model. So basically, I think we have this intuition that we don't need that many data. We probably need data on the order of n being the number of resident species. Okay, so then we did more simulations. So in these simulations, we generated simulated data, or just say fake data, using the model, Log-Terra model. And then we can tune two things. One is the training ratio. The training ratio is basically the number of training samples divided by the number of species in the meta community. So the more training data you have, of course, you expect to have a better performance by the machine learning models, right? So here we use logistic regression, neural ordinary differential equation, and Warren Forest, and they in the simulated data, they all have some reasonable performance once you have enough training data. The threshold is like around 1 and 2. So basically, if you have 500 species in the meta community, then you need around 500 samples to have a reasonable prediction. But also we tune this C, which is the connectedness of the network. So basically, not all species have interaction, right? So if you believe the interaction network is sparse, then of course it's easier to make predictions. If you have more non-zero strong interactions, then you need more data to have a better prediction. So that's all very reasonable. Okay, so then we decided, okay, we should do some experiments to validate if this really, we can really do the data-driven approach. And so we use the stool-derived microbial communities. And the reason is that because we need at least hundreds of data samples, and you can't do that with mice, right? It's just too expensive. And also, mice is also different from human, right? So we developed this high throughput in vitro anaerobic culture. So basically, we grew the gut microbes in anaerobic chamber, where it basically filled with nitrogen, carbon dioxide, and some hydrogen. And then you can grow the microbes extracted from fecal samples in these 96 well plates. Okay, and the composition actually can mimic the in vivo composition to some degree, and they can actually can maintain the individualized composition. And also, if you actually put them back into mice, and people have found that it's the response to, for example, to pathogen invasion and to antibiotic treatment is quite similar. So in this in vitro model versus the mice experiment. So that tells us, okay, we can use this great high throughput system to collect our data. So in our data set, we use 24 different subjects, their fecal samples, and then we also had 12 different antibiotics treatment just to diversify the baselines. Okay, so in this case, we have 24 times 13, so it's roughly about 300 samples. So these 300 samples with 3 replicates, they were actually grown in this 96 well plates, and then we passage these communities. So every day we did dilution. We diluted 200 times and then transferred to the next day. Okay, so the whole experiment took about 2 weeks. And the first week, it's called like a steady state generation. So basically, because it started from the fecal sample, right? So we passaged it about 7 days. So it got steady state. And then we invaded the communities with a pathogen, for example, and then we see what happens for the pathogen, right? So this is the in vitro system. And so this is just to show you some intuition about how these different communities look like, right? So each column is a different species, and each row is a different sample. So they could be from different human subjects, could be treated with different antibiotics. And as you can see, some antibiotics treatment would remove or reduce the load of some taxa. Okay, and this is a number of coexisting species. So because we use the metagenomic sequencing, there is, of course, a sensitivity cutoff, right? So any species below 10 to the minus 4, we just treat them as not existing. And so for different samples, we have about 40 to 120 coexisting species. The median is around, let's say, 100. Okay, so this is a highly complex community, right? So 80 to 100 species, they coexist. So then I want to show you this key experiment where we introduced a pathogen called Enterococcus faecium. So it's a pathogen that often causes hospitalized the infection. So we introduced this species into different communities, and then we sequence the communities before and after invasion, the steady state, and see what happens. So what happens is that remember I told you there are two qualitative outcomes. One is permissive, one is resistant. So 60% of the communities, they are resistant, so meaning that when introduce this eFeces at 5%, then after 7 days, it's gone. Okay, they cannot colonize. And for the 32% of the communities, this pathogen colonized, and but they have huge variations. So some after colonization have, let's say, 1%. Some is like 1 in 1,000 or 1 in 10,000, right? So they have different levels of steady state. And if you compare the diversity of the resistant versus permissive communities, as you might expect, the resistant communities have a higher diversity. So when you have more commensal species there, it tends to be more resistant. Okay. But if you plot the community composition, then there's definitely overlaps of the resistant versus the permissive. So it's not quite obvious if you can predict it, right? So, but then we feed the data into the machine learning model, like I mentioned. So I just show you there's 3 different models, and the random forest had the highest area under the ROC curve, and it's 0.86. So this meaning that if you have enough training samples, then you can qualitatively predict whether Enterococcus faecium can or cannot colonize in this community, right? And another thing that we wanted to ask is that, say we can predict, okay, a foreign pathogen can colonize in our gut or not, can we identify the key taxa for the resistant, right? So what species in my commensal communities are actually combating this pathogen, right? I don't really know. So we can do a thought experiment. So we can, because we already trained this model, right? So basically this is trying to figure out the feature importance. So we can remove the feature, right? We remove the resident species and see what happens to the predicted outcome. So we call this colonization impact. So basically this is a thought experiment to compute the colonization impact. So in simulated data, we find that this so similarly, meaning that using GLV model where we know exactly the interaction strength R, we find that this colonizing impact is highly correlated with the interaction strength. So then we will look at the real data. So the eFeces colonization data, what we found was that the colonizing impact is mostly negative. So it could be positive or negative, right? So there we have no bias, but what we found was that a lot of them are close to zero, but some are quite negative. And this from left to right actually is the most negative colonizing impact, meaning that they are really strong inhibitors of eFeces. And this one really stands out. So this species is called Enterococcus faecalis. Okay, it's in the same genus of Enterococcus, but a different species. And so we decided, okay, this seems to be top 1 inhibitor, so we should validate that. And so we introduced E. faecalis before eFeces or together with eFeces or after eFeces. In all these scenarios, what we found was that in our system, it would greatly reduce the abundance of eFeces, the red species. And these different red colors are 2 different strains, just to see whether it's generalizable. So what we found was that, and the B1 to B8 are different communities, right? So in different communities, we found that E. faecalis is a very strong inhibitor of eFeces, right? So when it's present, eFeces seems unable to colonize. And this is actually, we look back into the different subjects, human subjects in our samples. So eFeces in red, E. faecalis in blue. You find actually for subjects that start with a high E. faecalis abundance, then eFeces almost always has a very low abundance, right? So, and we look at the correlation of these 2 species in human samples. These are natural samples, not in our experiment. So we have a 120-person cohort in our Seattle Institute, and also we have a look at a human cohort with thousands of people in Israel, in UK, these different cohorts. We find that these 2 species obviously have a very negative correlation. But yeah, these are just correlation. But in our experiment, we find that we validated that they actually have a very strong inhibitory relation. Okay, so just to show you a little bit more, I think so. You might wonder why we picked this ephesium, right? So, but we actually tried different species. So in our experiment, and one species that you might have heard, it's called Echinacea mycelium. It's a probiotic.
Yeah.
Some people call it AKK.
Mhm.
So it's a second generation probiotic strain. And we also find that for vancomycin treated samples versus control sample, there's different variations of the colonization. So we decided, so this is more a small scale pilot experiment. And we find that some species have very large variations in their colonization. So we also did this for AKK, and so the data is interesting. So because in this case, about 90% of the communities, they are permissive. So AKK can colonize. Only 6% are resistant, right? But for the communities that this AKK can colonize, there's a bimodal colonization. So some are actually very permissive. So the AKK can reach abundance like about 10%, but some are less permissive. So it's 1 in 1,000, right? So this high or low permissive groups. And we can try to predict qualitatively which group they are, or we can even try to predict the post-invasion abundance in a quantitative way. So also in this case, we find that random forest seems to have the best performance. Okay, so anyway, this is just to show you that the data-driven approach is generalizable if you want to study other species of interest. Okay, so to wrap up this part, I think we propose this approach where you can predict the function of complex communities without knowing the magnetic details, right? So if you have enough samples on the order of the complexity of the system, then I think it becomes predictable. And you can also just to look to figure out what are the important features, right? It's not just a black box model where you can identify some key microbial taxa that mediate the function, in this case, the colonization organism. Okay, so just a brief outlook for this approach. I think we start with collecting data. We observe, okay, there's variations among different communities, and try to understand what commensal taxa, in this case, Ephecalis, play important roles in folk resistance. And then you can engineer the community, right? So for instance, if you want to improve the resistance, then you just introduce some strong inhibitors, right? Which might be lacking in your gut. In another scenario, if you want to facilitate the colonization of probiotics, then you might introduce some species that help the probiotics, right? And eventually, I think we're not quite there, but if we have more and more data, we might be able to design microbial consortia once we know the community to function landscape, right? So this landscape, I think, is if you know protein evolution, then people like to use protein fitness landscape, thinking about the proteins are evolving to climb uphill to have a better function, say the enzyme function, for example. So in this case, I think we really need to know the community function landscape because if we want to design a community. Okay, so that's the second story. So for I think I would just, because I don't have that much time, just very briefly introduce the two other stories. Okay, so very quickly, okay, maybe use five minutes. So we developed some novel tools. One tool is to understand strain level composition given the metagenomic data. So we call it strain panda. If you like the pandas, so you can just very easily search. It's available on GitHub. So the question this tool solves is how can we know if in your metagenomic sample there are multiple coexisting strains? So these strains, so if you have E. coli, they can have different strains of E. coli. Some are pathogenic, some are probiotic, right? So the different strain, they have of the same species can have very different gene content profiles that which would influence their phenotype, like metabolic capacity, colonization, and pathogenic capacities, right? So this tool that we developed and published in 2022 on iMeta, which is a new journal on bioinformatics and metagenomes. And so it's actually, we use the non-negative matrix factorizations to decompose the metagenome pangenome. And so we can have both the string level composition as well as the gene content profiles. So I just skip this the details to show you example. So these are simulated data which we just show that it's very accurate. And so let's get back to say the infant data. So we have, like in this data set, each infant has 3 different time points. So right after they were born, 4 months and 12 months. And we found was that for this probiotic, Bifidobacterium longum, we inferred there are 3 different subspecies. And well, after breastfeeding, subspecies 2, it went down. So if you only have species level information, then you don't really know the subspecies, right? So once you have string level information, then you find out actually on string level, there's ecological succession. Subspecies 2 went down. Okay, so why? So I think what we find out with, subspecies 1, that the abundance went down. So what we realized was that they have different gene content profiles. So for subspecies 1, they have multiple genes that are involved in human milk oligosaccharide utilization, HMO utilization. So that's the carbon source from the mother's milk, right? So once you stop breastfeeding, then its fitness advantages come. Okay, so then its abundance went down. So I think this helps us understand at the subspecies level how the strains are competing for resources. And this, if you apply our tools, then you can apply to gut microbiome, plant microbiome, just to figure out at the strain level which species are present, which strains are present now, what's their gene content profile. So using this non-negative matrix factorization algorithm. Okay, so lastly, I think I just show you another study that we published last year about the spatial composition, spatial profiling of microbiome. So sequencing basically can tell you, okay, in a well-mixed case, what is the composition of the microbial committees? But a lot of times we know that microbes don't actually are organized in a well-mixed manner, right? So, for example, on leaves, in our oral cavity and in gut, they have very interesting spatial organizations. Okay, a multi-species biofilm, and they organize in such a way because there's environmental heterogeneity, because there's, for example, the oxygen gradient, there's host metabolites, there's interspecies interactions. So there are many factors play a role, and we develop a new method to image the complex microbiome, because for traditional fish methods, you can only label a limited number of taxa. So for example, you have 5 fluorophores, you can only image 5 species at a time. If you want to image 100 species, that's not feasible. So we develop a sequential fish method. So basically you do multiple rounds. We can do 10 rounds, 20 rounds to image more species, and we assign each species a color barcode, and we introduce some Hamming distance among the barcodes to correct for errors. So we call sequential error robust fish. So you don't need to know the details, but I just show you an application example. So we image the plant roots. So as I mentioned in the very early beginning, there's a lot of microbes living in the soil and in the rhizosphere. And why? Because plants actually secrete a lot of their photosynthetic carbon source to the rhizosphere. So as you understand, they're trying to modulate the bacteria. They attract the bacteria, right? The good bacteria to help them. Okay, so we did in our laboratory where we co-culture the Arabidopsis, which is the model, very easy to culture plant with the rhizosphere bacteria. And then we take these samples and fix these samples for imaging. And what we found was that in the response to this plant root metabolite, there's a shift in the composition. So actually, this is not sequencing. This is by imaging. Okay, so you image each sample and then count the number of cells for each different taxa. So they're very clear shift. So this is to show you, I think, a very intuitive understanding. So when in response to different plant metabolites, you definitely see there's a shift in the colonized microbes and also in their spatial organization. So these pseudocolors correspond to different species. And this probably the more clear pictures. I just pick one root, and because we did this at a single cell level, so you can zoom in and you can see, okay, these are different microbes, right? And then they definitely have some kind of patterns, both at the local level and micron level, as well as the different compartments of the roots. And we can perform a quantitative analysis indicates a linear dipole analysis, where basically you're trying to calculate the probability of a dipole that hit two different bacteria. And if this number equals 1, it means it's randomly distributed. If this is larger than 1, it means there's aggregation at a certain distance. So we image the root at different compartments, and if you zoom in and then calculate these, the function that I just mentioned, so you can see we're at a distance, I think around 30 micron, you do see a lot of aggregation. So it's not just random. It's not random, it's aggregates. And you can calculate also in these multi-species aggregates, what is the probability or what is the association among different taxa. So some taxa seems to like each other, right? And some are not. Okay, so you can get a lot of spatial information using these the highly multiplexed spatial profiling methods. And we're now applying this to other samples like the gut microbial samples to understand the spatial distribution of microbial communities. Okay, I think that's all. I just summarized the work I mentioned. So we really need to understand microbiomes from a system biology perspective. I think the quantitative and mathematical methods help a lot. In the dynamic response, the two stories that I spend most of my time on. One is, I think, a white box approach, where you started with a parametric model and then you try to understand the interaction terms. And also another is a deep learning model, where a machine learning model, where you just have a black box model and then try to predict the function. And also, I think a lot of times for microbiome studies, we really need new tools. So for example, at a strain level, strain resolution, we need to understand whether there are multiple coexisting strains. And also, we need to go beyond sequencing to understand the spatial structures. And that's all. I want to acknowledge my lab members and collaborators, and also just you're welcome to contact me if you're interested in doing a postdoc or applying for some PhD positions in China. So this is Google map. So Hong Kong and Shenzhen right next to each other. This is roughly where our lab is. So it's very close to Hong Kong, just actually half an hour by high-speed rail. And this is our new campus, where we have a lot of automation robots to do the experiments. So even if you are not a biologist, if you're comfortable with programming, you can probably handle the wet lab. And also, this is our old campus, and these are some just nature outside the lab. So, okay, thank you. Sorry for a little bit over time. Yeah.
No, it's
Okay.
totally fine.
Thank you.
Thanks so much, Lei Dai. So everyone, let's first thank Dr. Dai. It's a Q&A time, so please feel free to unmute yourself and then ask your questions. Okay. And then let me do first, Dr. Lei. So at the last part, you talk about that microbiome is pretty localized, right? But but the first section, a lot of people use the still that looked up Volterra type ordinary differential equation model to infer the network. So to me, sort of
Mhm.
two appears to be conflict a little bit because ODE assume that homogeneous distribution, right?
Yeah,
Yeah.
yes. So talking about how to reconcile these
Mhm.
approaches. Yeah, I think if you only have sequencing, then you already assume like it's a well-mixed sample, which a lot of times, for example, we have fecal samples. Fecal samples is very close to the colonic region, right? So but it's yeah, so it's only one part of your gut, right? So it's okay to some extent to use that region to represent the entire gut, but obviously it's not quite accurate, right? So I think to a lot of microbiome studies, people start with sequencing because it's easier to collect samples to get data, but we must keep in mind that you only look at some region, and also within that region, there is also heterogeneity, but you just ignore that, right? So if you ignore all these heterogeneity and then let's say the within host heterogeneity, just think about it, a well mixed like a bioreactor, right? These different species, then you can also get some understanding, right? So for example, in the gut, you have lumen or you have the mucus, right? So in the lumen, it's more or less like a well mixed case, right? So it's okay, but once so you just be cautious about when the assumption breaks down. I think it's okay to use the differential equation models to understand, for example, like at different time series, you see drastic shifts, right? It's okay because you know that species dominant, right? So even without knowing the within host heterogeneity, it's a dominant species, so it's okay, right? So, but other times, right, when you actually think about, okay, what happens at a micron scale, right? And then we really need to actually, which I think the spatial microbiome field is just emerging, because a lot of times the observation happens after you have the appropriate technology, right? The 20 years ago, we even don't have sequencing data, but now we start to have more and more imaging data, and then we'll see if when it deviates from the well-mixed case, or yeah, so we don't really know that much. Like even for the root sample, we had this observation, but we didn't quite understand it, right? Why at different compartments there's a different set of microbes residing on the compartments. But then I think I think half a year or a year after my publication, I was glad to see there was another study. They find that there's, for example, a single cell sequencing of the plants, and then you see that at different sections along the roots, they have different expression of their receptor or something, and then the plant basically they secrete different carbon sources or sugars, and that would attract, I think, different microbes, right? So eventually, I think you will reach a better understanding. Right.
Mhm.
If you treat it as a well-mixed sample, you get some understanding. But if you have data for the spatially resolved data, then you have a better understanding. Yeah,
Okay.
that's my take.
Okay. Thank you. So yeah, any other question from the audiences? Yeah, please feel free to unmute yourself and then ask a question.
I have a question. So,
Yeah, please. Yeah, take the
maybe
one.
maybe I think I got lost, but when you use machine learning to, are you inferring the network which follows the generalized load cover data?
No, I'm not trying to infer the Lotka network. I'm just trying to make prediction from the composition to the resistance. Basically, in this case, it's the new steady state. What is the abundance of the invasive species, right? So I'm trying to predict that without knowing the interaction terms. But I think your question probably stems from the one slide I showed about how can I predict which species are inhibitors, right? So in this case, what we found was that if we do this thought experiment, like we train the model and then we already have that mapping between community composition and the outcome, colonization outcome. So we remove each species and see how the outcome changes. And this colonization impact value we calculated is actually related to interaction strength in the GLV model. Yeah, so that's in some way but I would say put it more precisely. I think if you go to read Professor Yang Liu's papers, they have this definition of direct versus indirect effect. So basically, if you have a network, right? So let me see, have that network somewhere. So there are two types of interactions. So let's say C. cindens, this number 4, it directly inhibits C. difficile, number 2. Okay, but can inhibits this 11 and then 14 and then some go all the way to this. So if there's an indirect network effect. So I think what the colonization impact is actually indirect plus direct. So it's the overall network effect on this. I'm not sure if I convey this clearly, but I just want to tell you that I'm not trying to infer the coefficients, the interaction coefficients, but more like what is this species doing on the other species in the context of all other species. Yeah. Okay.
Okay. Okay. Thank you.
Yes. Any other question?
Thank you for
Yes.
nice talk.
Yeah.
And I have one question. In my thought, you extract some abundance factor from the metagenomic, metagenomic sequencing data, right?
Yeah.
Yeah, and I wondered that. So, what is the metagenomic data first, and from this, after you make model, you predict the colonization outcome, right? Using that abundance factor, right? So is there any correlation between the community structure and colonization outcome?
Between the what structure? Between the
because you use the community structure as an input and the colonization outcome as output, right? Is there any correlation between them? Correlation. So if they have a similar community profile, so then they have a similar outcome, right?
Similar, well, depends on how you define similarity, right? So I think to some degree, your statement is correct because similar community structure. In this case, this structure just means composition. Okay, let's say these two vectors with some similarity metric, like a lot of times we use Curtis distance or use other distance. So if the composition is similar, they probably have a similar function. I think a lot of times this is true, but I think it depends on. For example, in our case, we find that for Endococcus faecalis, it's a strong inhibitor. So if I just miss this species, then it could have a huge impact on my function. But if I miss some other random species, it doesn't really have an impact, right? So I wouldn't say that. So it's related. A lot of times you can say that similarity in composition might lead to similarity in function, but it may not always be true. Yeah, so that's I think my answer to your second question. The first question is how do I get the metagenomic sequencing? How do I get the vector? Is that the question?
Yeah, because I'm currently dealing with the single cell sequencing data, so I know that data's property, but compare with that. So I want to know the difference.
I see. It's actually very easy to implement the metagenomic sequence analysis. So basically, put it very simply, so you start with DNA extraction, and then you prepare for the next generation library sequencing, and then eventually, when you got the sequencing data, it's actually just so basically it's some reads. Each read is about 150 base pairs extracted from a certain microbial genome, right? So then you count the reads just to figure out if you have 100 different species, right? And let's say the species number 1 has 10% relative abundance, then it probably has the 10% reads correspond to this species. Okay, that's very simple calculation, but you need to account for marker genes, genome size, other stuff. So, but there's very well-known tools like Kraken-2. We also use some other tools. So basically, you process the DNA reads to calculate what is the proportion of each species in this entire sample, right? So eventually you get a proportional vector, right? So what I said was absolute abundance. How do I get absolute abundance? What we did was that we used quantitative PCR. So because each species have a marker gene called 16S ribosome RNA, so we can basically use the total copy of 16S genes to estimate the total number of microbes in that sample. Because for some sample, you could have 2-fold or 10-fold difference in the absolute number of bacteria cells, right? So that also matters sometimes, but not always. Yeah, so a lot of times people only deal with compositional data, just ignoring the total absolute abundance. That's also fine, but yeah, okay.